happy to introduce Paul Janmey, who's a university professor at UPenn, so not from far away. He's basically the person to go to if you want to know about how the cytoskeleton works and how the actin dynamics inside the cell govern all sorts of processes in the cell. And he's one of the rare breeds of scientists who's actually fundamentally interdisciplinary. So that means he has a physical sciences background in his own education. Then he went into biology and biomedicine and even worked in a hospital and figured out what the interesting problems in the biology and biomedicine world are. And then he decided to phrase those questions in a physical sciences kind of perspective. And so he's special in that way because he says that physicists have been kind to him. And in particular, he was working in collaboration with Tanaka in Harvard when he was at Harvard, and he was working with Eric Zachman when he was in Munich. And he said that both of these fathers in physics were very keen to introduce him to the world of physics, and ever since then he's been collaborating with physicists in understanding how the cytoskeleton works. And so I wanted to just emphasize, or at least introduce the history of Paul being in this subject area, which is that he says he had a lecture when he was being educated in polymer science and how polymer gels work. And for one of the exams, they had a question saying, so how does this polymer gel behavior compare to that of the cytoskeleton inside the cell? And he thought, I could do that. I think I know how to do that. And so several years later, I don't know how many, he has taken that subject quite some way along that path, and that's what we're going to hear about today.
Great.
Thank you.
Great. Thanks very much for the chance to come here and show you some things we've been doing recently to try to look at the kind of the soft matter physics of soft tissues. And so I want to tell you sort of 4 short stories, really 3 stories, but one big question. What we're really interested in is trying to understand what are the molecular structures and what are the mechanisms that dictate the stiffness of different tissues in the body, because as it turns out that seems to be an important biological control mechanism. And our initial attempts were to try to make comparisons, to try to make some sort of contact between that tissue mechanics and the mechanics of networks made out of the semi-flexible biopolymers that make up most of these tissues. And I'll show you to the extent to which that seems to work and where it seems to fail. And in particular, recently we've been very interested in looking at the elastic properties of materials within the body under the kind of the relatively large deformations and stresses that these tissues are designed to experience in vivo, and to look at some of the responses of putting pressures or stresses on the materials themselves. And in doing so, in trying to put more physiologically relevant kind of deformations on the materials, one of the surprising, or maybe not surprising, things is that this parallel, or this attempt to make some sort of connection between polymer networks and soft tissues has really serious limitations. And there are many, in particular, the responses of these materials to uniaxial compressions is exactly opposite. And we're trying to understand why that happens. And our approach to try to do this is try to make these purified, simple polymeric systems more and more messy by populating them with internal stresses or with particles to make them look more tissue-like, and I'll show you how far we've gotten. So the work I'm going to show you is largely done by bioengineering and cell biology people in the lab. I'll show you their contributions later, but Jeff Byfield, Li Kang Chin, and Kasha Pagoda are all physically trained people, and Van Oosten is a bioengineer in the lab at the moment. Okay, so here's the motivation. And I'll try to keep the biological motivation short so we can get to the kind of the material science as soon as possible. But the reason we're interested in this from a biophysical, or from a sort of physical point or physiologic point of view, is that soft tissues in the body have surprisingly well-defined elastic moduli. That is that, and I'll show you a couple of examples later, but if you take tissues from people of the same age and the same state of health, or animals at the same stage of health, they have a remarkably well-controlled mechanical property of their tissues that depends on a lot of factors, most of which are unknown, but that changes a lot in disease. That's why I think people are interested in this, because, for example, tumors are often, but not always, stiffer than the tissue from which they came, and fibrotic livers are stiffer than normal livers. And so these kinds of mechanical things are extremely useful in diagnostic devices, and that's why something like physical palpation works in diagnosis, but more sophisticated imaging methods take advantage of changes in either density or elastic properties of tissue to tell you where something has gone awry. But why they are stiffer is kind of or what determines this elastic modulus to begin with is a little bit mysterious, because the cells within those tissues do not have genetically pre-programmed stiffness. If you take a cell out of a tissue and culture it under different conditions, its own stiffness can change over a very large range depending on what you do to it. So something about its homeostatic state inside the tissue has made the larger scale thing very well controlled, but the individual ingredients not so well controlled. So we're trying to understand how that feedback works. And the reason for that is if we understood something about the physical principles that nature uses to make a liver or a brain or fat or anything as stiff as it is, we might be able to get some idea of what's going wrong when a disease kicks in. So before I get into our data, I want to show you a bit of kind of prehistory, because it's sort of interesting, and it's sort of chilling how much has been done that we're just repeating. And in particular, I want to tell you this, a little story about this guy, William Seifritz, who was a botany professor at Penn 100 years ago or so. And he is, I think, the first person to do microrheology on any kind of biological system, or even on a gel. So he was a botanist. He did a kind of not exactly a postdoc, but he went to work with a colloid physicist, Herbert Freundlich, in Berlin in the 1920s. And they built this beautiful electromagnet sitting on top of a microscope. They embedded magnetic particles in cells and yanked them around with a magnetic field gradient that they could generate at the tip of this electromagnet. And from this kind of work done, almost 100 years ago, they figured out that the cytoplasm, the interior of these cells was not a simple liquid like most people thought, but that had a lot of the properties of a solid. And the U-tube has a beautiful collection of weird stuff in it, as I'm sure you know. And among the weird things that you can find is this, I hope this plays, this movie made in the 1950s in which Seifritz explains how they did what they did. Let's see if this plays. It's just wonderful to watch.
I want to tell you about a very lowly form of life, a slime mold, the present-day counterpart of the primordial ooze that appeared on Earth many, many eons ago. The descendant of this primordial ooze, the substance which gives life to all plants and animals, we call it protoplasm. Now let us go to the woods and collect the protoplasm of the slime mold.
Wouldn't it be great to do this?
You probably find it growing on an old stump, such as this one. Or if the first stump reveals nothing, you search for another.
All right. I'll stop it here. You'll have to find out what happens next. So in looking at things like this primordial ooze, and by looking at the interior of cells, these guys were able to conclude some really interesting fundamental properties of the material inside the cell. One of which, in addition to simply showing that it has solid-like properties, they also were really very well aware of the fact that they could relate the physical properties of the cell interior to the metabolic state of the cell. In particular, a state of high metabolic or physical activity means low protoplasmic consistency. Or in other words, low viscosity is coincident with an active state. So they already knew at that point that these things were materials out of equilibrium where particle motion was driven in non-thermal but random looking, what looks like Brownian motion. And in many ways, 90 years before, a slew of recent papers, such as this beautiful paper showing that the cytoplasm, even of bacteria, has these glass-like properties that are fluidized by metabolic activity. That's exactly the same thing that these guys were onto in the 1920s. The other kind of landmark in this sort of early thing, as you may know, is the thesis work of Francis Crick, who 30 years later basically redid these experiments of Seyffert's and wrote this really nice but actually remarkably snarky paper where they kind of grudgingly admit that, oh, these guys shoved some nickel spheres into gelatin and that these experiments are unobjectionable, except, of course, for blah, blah, blah. But the important achievement of this early paper of Crick and Hughes was that they were able to put a quantitative number on the elastic modulus of the cytoplasm, again from embedding magnetic particles within a cell and pulling on them with an electromagnet to do kind of micron-sized rheology of the cell interior, and concluded that the inside of the cell has a modulus of rigidity of about 100 dynes per square centimeter, 10 Pascal. That's probably lower than most people would think it is now, but it's kind of in the right ballpark. But one of the conclusions of this paper is this really remarkable thing that we would plead that the evidence we've presented for elasticity in the cytoplasm will not be regarded as evidence for some cytoskeleton in the cell. So that's probably not where most people who work in this area are going, but there's now lots and lots of evidence that there is a cytoskeleton in the cell.
What is cytoplasm?
What's cytoplasm?
Yes.
So what these guys meant by cytoplasm, and I think what Seyffert's meant by protoplasm, is the region of the cell outside the nucleus and inside the outer membrane of the cell. The entire area in there, which is now demarcated in kind of modern jargon as lamellipode, lamellipodium, cytosol, and this sort of thing. This was all kind of lumped together as cytoplasm. So the parts of the cell that look optically clear in a microscope, but that are clearly not the dense nucleus, but inside the membrane of the cell, that's all lumped together as cytoplasm. And the consistency of what you measure depends a great deal on how deeply into this material you put the magnetic particle. So that's more or less what they meant by it. But in fact, as microscopes have gotten better and better, and as people have looked more closely, it's pretty clear that both the inside and the outside of the cell is just littered with these large polymers. So I show you first a kind of larger scale picture of the kind of polymer networks that permeate tissues. So these are these kind of flat objects or cells, they're fibroblasts, and they are crawling through a network of polymers made from this protein collagen in a wounded, I think it's a lens, in an eye injury. So a lot of wound healing and even normal tissue development is driven by cells that are embedded in these kinds of networks, where the characteristic mesh size here is a few microns. These cells are maybe 10 to 50 microns in size, and the mesh of the network is much bigger. The other thing here in the colorized electron micrograph by my colleague John Weisel is a blood clot. It's a whole blood clot. So this is pretty much what it looks like when you cut yourself. The way you stop bleeding is that there's a very rapid polymerization of a different polymer, fibrin, into these networks, which again have mesh sizes on the order of a few microns. These red things are red cells, so that's about 5-6 microns in diameter. They're trapped transiently inside this polymer network. These little purple things are platelets, which are much, much smaller cells, but they're active in the sense of when your blood clots. These things are also enzymatically activated. They will send out pseudopodia, little arms, to attach themselves to the fibers and then contract it. And applying this internal contraction is a big part of making a blood clot. Yes?
I know this might be off topic, but what is it that initiates or starts this polymerization?
So what starts the polymerization in the blood clot is this insanely complicated set of coupled enzymatic reactions. So there are circulating enzymes or zymogens that are inactive enzymes to begin with, but if you cut yourself and expose materials to the blood that ought not be there. So for example, if you break a cell or if you break a tissue in the cell, one of the most active things that happens is the phospholipids on the inside of the cell, which are negatively charged, show up on the outside of the cell, which ordinarily doesn't happen. So the proteins that are floating in your blood are not used to seeing a negatively charged surface. If they see a negatively charged surface, as would happen if you disrupt the membrane, then surface active proteins stick to that, locally unfold or get activated on that surface and initiate a sequence of enzymatic reactions that lead to cleavage of inhibitory parts of a circulating protein, fibrinogen, which rapidly polymerizes in this kind of polycondensation reaction very quickly to form this kind of network. These collagen networks are made in a much more slow and ordered way. Cells secrete the molecules that make them and polymerize them, and these networks are, depending on what part of the body they're in, they're made to last. So there are collagen networks that last for years. The proteins of the blood clots are made and dissolved on a time scale of days. But this is hugely rapid and kind of basically random polymerization. This is much more directed. Okay, so the inside of the cell also has these kinds of objects in them, but with a much different length scale. So here's a electron micrograph of the top of a white blood cell, a macrophage, where if you just lift it off the membrane and peeked at what's inside, what you see immediately underneath the cell membrane is this polymer network, mostly made out of actin filaments, with a mesh size on the order of a few hundred nanometers rather than a few microns. The filaments are thinner. They're still stiffer, stiff enough so that they look relatively straight on the length scale of the mesh underneath them, and there are in a variety of different ingredients that make up the so-called cytoskeleton that Crick didn't believe in, and then fill the space. So part of the elastic character of the cytoplasm is driven by the fact that these polymers are everywhere within certain regions of the cell. So if you get a bead embedded in this, then it will have a very non-viscous looking reaction to applied forces. And its thermal fluctuations will be very much damped. So if you see any fluctuations, that's almost for sure a sign of active driving. Okay. So these are the ingredients that give rise to tissues, and as I mentioned before, one of the things we've been really interested in is what determines the stiffness of different organs or tissues, and what happens when that stiffness changes. And there are lots of examples of stiffness changes during the development of fibrosis in the liver, the elastic modulus. In this case, it's the shear storage modulus, so the elastic response measured in shear on about a second time scale in a very small deformation. It's about an order of magnitude bigger in a diseased liver than in a normal liver. Same with the development of malignancy in the breast. The breast tissue becomes much stiffer than it is either early in development or even in the pre-malignant tissue. And you can tell the difference by a variety of physical forces. I won't belabor the point, but there's lots of other cases in which the stiffness changed. But I do want to point out a little bit of evidence of how well controlled this is. Here's an experiment done by my colleagues at Penn, who have generated a transgenic mouse that is doomed to get atherosclerosis by knocking out a protein called ApoE. But in studying how this happens, we made some physical measurements of the aortas from wild-type mice and many mice, and you can see how small the variance. This is the standard deviation of a bunch of measurements. So whatever it is that determines the stiffness of the large blood vessels of this animal, it's very well controlled. And if you just make this genetic deletion, the blood vessels become much stiffer. And even before the animal is sick, this vessel has become stiffer, and it will very rapidly become inflamed and lead to deposition of stuff that will eventually give the animal heart disease or cardiovascular disease. And what we've been chasing is this idea that the stiffness change itself is not a consequence of the disease, but it might actually be a driver, and that's why it would be interesting to look at it. So I want to show you one example in which has motivated some of the kind of the physical studies we've been doing, because Kasia Pagoda, a student in the lab, was interested in whether this kind of scenario, that development of cancer leads to stiffening of the tissue and that may cause the cells to misbehave, whether that also happens in the brain. Because the brain is a really interesting organ. It's very, very soft compared to other tissues. It doesn't have these extracellular polymers I showed you before, these collagen networks. And so the question is, does the tissues get stiffer all the time? Here's a reason to think that the manipulation of organ stiffness has some consequence in biology, because we can do experiments ex vivo in which or sort of ex vivo, in which we cause something like a disease process to happen by, for example, intoxicating rats with carbon tetrachloride that will give them liver fibrosis, and then take out livers from them several days after the intoxication, measure their stiffness, again by looking at the shear modulus, and sending slices of these things to a pathology lab to look at whether the tissue looks diseased or not yet. And one of the interesting things is that after you've already given the signal for the tissue to go awry, it becomes stiffer before you see any evidence of cells misbehaving. So the stiffening process, which we think has to do with a chemical cross-linking of these extracellular networks, that stiffening process precedes and might therefore cause the cellular dysfunction, which eventually will lead to liver failure and that sort of thing. Okay, so that's the idea. And you can kind of reproduce a pathologic response in cell culture simply by taking a cell type. In this case, it's a liver cell, a hepatic stellate cell. It's one of the cells that will become overly activated in fibrotic disease. And if you take it out and culture it instead of glass or plastic, the way most cell culture is done, if you culture it on a soft material that is as soft as a healthy liver, the cell has this kind of, not very photogenic, but a phenotype that it retains in vivo. And if all we do is change the stiffness of the material around it, we can spontaneously activate that cell to become stellate and to start secreting the kinds of molecules that are associated with fibrotic disease. So this is at least evidence that changing nothing but the stiffness will cause a lot of cellular responses. So that's the reason to think about, sorry.
The substrate is chemically the same, but it has a different stiffness.
Yeah, it's chemically the same. It's chemically identical. We've just changed the stiffness of the material underneath it. And I'll show you, I think, in a cartoon next how we do this. I hope it's the next cartoon. Yeah, it is the next cartoon. So the way we do these kinds of measurements is, rather than culturing cells on glass or plastic, we put on top of them an inert hydrogel. It's usually made out of polyacrylamide, the same stuff that people do electrophoresis with, or other gels. But the main idea is that this blue thing is a little mattress, an inert mattress. It's not drawn to scale. It's actually enormously fat. It's 100 microns or so fat. And it is a polymer to which the cells cannot stick. So they do not see the polymer themselves. And we can change the elastic modulus over a pretty long range by either changing the polymer weight fraction, which we try not to do, but more by changing the cross-linking concentration, which is much easier to do. So the texture that a cell would see from the top, or the kind of the mesh size of the polymer network, is relatively unchanged. We change the elastic modulus by changing the covalent cross-linking density. And so we can tune these gels to be over most of the range of stiffness I showed you of these soft tissues. And then the trick is to get the cells to adhere to it. We covalently link to the top of this hydrogel surface specific molecules to which the cell has a receptor. If we do not put a molecule to which the cell can stick that mimics its environment in vivo, the cells just bounce off the cushion. You have to give it something to which it can specifically adhere by integrins or some other transmembrane protein. So we think what we're doing is keeping the surface coating identical because we can see it and we can quantify how much of the adhesive protein we've put on the surface, and we just change the stiffness by putting it underneath. And so we don't have to do anything chemically. Just changing the elastic modulus, the stuff underneath, will give rise to all kinds of phenotypic changes. And one of the most pretty changes is what happens when you take muscle cells from, in this case, neonatal rats. And in the body, the muscle cells are elongated. They have these nice striated things, characteristic. This is what the cell used to look like when it was in the heart, and then the heart was all blown up. And if you take these heart cells and culture them on very stiff gels, something that is stiffer than the heart muscle from which it came, you get cells that are alive, or if you put them on glass, you get cells that actually are quite beautiful to work with, but they do not have the function of a muscle cell. If you put them on gels that are much softer than the tissue from which it came, which in case of heart is just in this area, 5 or 10 kilopascal, if you put them on very soft adhesive surfaces, again, they will stick and they will live, but they will not differentiate into something that, they will not reacquire the morphology that they had in the tissue when you took it out. If you put it on something that has the same stiffness as the material you took it out, it spontaneously regains this kind of phenotype. And so we haven't done any chemical stimulation to get it to do this. We haven't patterned the surface. We just put it on an infinitely sticky surface and ask it to do what it wants. And if you get the mechanical environment right, it will reacquire this normal phenotype. Yes?
Does the same thing happen when you have multiple cells on the surface? So instead of taking one muscle cell, if you had 3, would you see the same effect?
Mean 3 next to each other? Yeah. Yes. We see a whole field of these things. What is really interesting and relatively unexplored is these muscle cells also want to make attachments at the ends to another neighbor. And the extent to which they can make attachments to each other also depends on how stiff the material is underneath it. So if you put them on very stiff materials, they have a very difficult time making attachments with the right geometry. And if you put them on these soft things, they can do it much better. What's less clear is what happens if you now stack one cell on top of the other. So this is what we've tried to do with brain cells to see whether if we take a cell from a tissue which is very, very soft, not tens of kilopascal, but hundreds of pascal, like the brain, do these cells also care about stiffness or not? So the cell types we've been looking at are not normal brain cells, they're glioma cells, they're cancer cells from a really nasty type of tumor. And there are many different kinds of these tumor cells, but they all retain the ability to tell the difference between soft materials and stiff materials. It happens that for these cell types that come from the brain, they can tell the difference between a few hundred pascal and a few thousand pascal, but by the time you get to 5 or 10 kilopascal, they might as well be sitting on glass. So for these cells, 10 kilopascal is infinitely stiff, but they can still tell the difference between very soft and stiff. Okay, so this is work that Kasia started, and this finding that these kinds of brain cancer cells can tell the difference between soft and stiff might be important because other cancer cell types have been shown to really proliferate and really do better if you put them in stiff environments that might mimic the tumor environment. And so the idea is that as tumors stiffen, the malignant cells within them are in an environment that's more conducive to their further proliferation. Okay. So we asked whether, in fact, brain tumors, I'll skip these because these just quantify what I showed you. It's a beautiful graded response. If we look at something like the spread area of the cell is a function of the stiffness of underneath it. It's pretty nicely regular. These are standard errors. There's a pretty big variance, but there's a very clear graded response of almost any metric we have of the cell mechanical features as a function of the mechanics underneath it. So these are exquisitely mechanosensitive cells. So therefore, it's an interesting question to ask whether the brain, a piece of tumor, is stiffer than a normal brain, and whether that gives a selective advantage to the cancer cells in it. And so the way we've measured these is we've gotten about 14 surgical samples now from brain tumor patients, and they're very small samples, so we have to come measure them by surface indentation with a 100 micron diameter needle or so, and from that we get some kind of measure of a compressional modulus that might have some relation to a Rheologic modulus, but in any case we can quantify an effective stiffness. And somewhat surprisingly, the stiffness of a real tumor is not any bigger than that of the normal brain. So at least in this case, either this hypothesis is false, that the environment, that the mechanical environment of the tumor is different from that of the normal tissue, or we're doing the measurement wrong. Yes?
Why are you comparing it to mouse brain?
Sorry?
It's human.
Yeah, we're comparing to mouse brain because we cannot get human brain. Although you're welcome to, but there's a literature from before that has shown that if you take a normal brain from trauma patients, there is some measurement of the mechanics of normal human brain compared to mouse brain. They're very similar to each other. So we take this as a surrogate because previous work has shown that there's not a dramatic difference. But we would have expected this to be kilopascal, not a few hundred pascal. Yeah, but that's a good question. So either this biological hypothesis is wrong or we're measuring it in an incorrect way. So we thought of two things. One, we are measuring this by very gentle indentations in a kind of linear elastic range, and probably tissues are undergoing very large strains. And work from a lot of labs from before that has looked at networks of these purified polymers like collagen and cytoskeletal things that I showed you before. These things are all characterized by this beautiful nonlinear elastic response in which their shear modulus is a very strong function of the shear strain. So the more you deform these networks, the stiffer they get. And there's theory from Kay Storm and Fred McIntosh and Tom Lubensky and others that have fit these data to try to get an idea of where this nonlinear elastic response comes from. If you look at something like a simple flexible linear hydrogel like polyacrylamide, its shear modulus is completely independent of the shear strain over any kind of reasonable range. But these stiffer biopolymers have this fantastic strain stiffening. Okay, so the brain, presumably the brain cells are made out of these ingredients, and maybe brain does this same thing too. And so we shouldn't be looking at the linear elasticity of tumors versus normal brain, we should be looking at the large strains. And I'll skip this over, and it's wildly disappointing. So tissues, as opposed to the networks of polymers, do not stiffen in shear deformation. They slightly soften. And so that's too bad. So that's not the explanation. So there's still no evidence that tumors might be stiffer, but somewhat to our surprise, actually a lot to our surprise, if instead of deforming them in increasing shear deformation, we put a small uniaxial compression on the sample first. So we take a disc-shaped sample that we're doing torsional deformation of, and we compress it a few percent. In this case, 40%, but it could have been 10%. We compress it a little bit uniaxially and keep it compressed, and now measure its shear modulus. Now it's way bigger. It still doesn't stiffen with increasing shear strain, but it does increase with uniaxial strain a lot. And this is not potentially irrelevant, because tumors and a lot of tissues in the brain, especially when they get diseased, they're subjected to fairly large pressure gradients. And so people have measured this a lot. In brain tumors, for example, there's about roughly 10 millimeters of mercury pressure difference between the inside and the outside tumor on a kind of a centimeter scale or a sub-centimeter scale. And so this corresponds to thousands of Pascal of stress in the direction of the pressure gradient. So presumably tumors are under, or at least the tumor tissue is under a compressional strain, and surgeons know this because they have to calculate what these pressure gradients are so that when they take the cranium out to do surgery, they know how much the brain is going to bulge out. So these pressure gradients may be giving rise to these kinds of deformations in the brain that cause the local shear modulus to change. And because the local modulus, the local stiffness of the environment, is what the cell responds to, taking the tissue out and relieving the pressure that it was experiencing at the time the cells are doing whatever they're doing is erasing the information that the cell gets in this disease state. At least that's the hypothesis. And so if we now look again at these tumor samples or normal samples and try to do, at least in a very limited set of experiments, measurements in which we try to measure the stiffness of the tissue as a function of the compression we put on it, glioma samples stiffen a lot more than normal brain. So something has changed in the architecture that might cause the interior that the cell actually experiences to become stiffer. Okay. So that's it about the brain. But I want to show you that this kind of thing is a fairly generic thing, and it has a lot of interesting mechanical kind of signatures. So if you look at the shear modulus, so this is the shear modulus measured at low strain in an oscillatory way. So we're measuring a very small amplitude shear deformation of a slice of brain, but we're measuring it under either static compression or static stretch. The shear modulus is now a very strong function of the axial strain, but only when you compress it. If you stretch it, it doesn't change. And it's a generic result. It has nothing to do necessarily with the architecture of the brain. It also happens in the liver. If you compress the liver, it gets a lot stiffer. If you stretch it, it does not. So there's some kind of interplay between axial deformation and shear resistance that we don't understand, but that might be really important. And it happens even more spectacularly in fat. So this is adipose tissue. If you compress it, the local shear modulus gets a lot bigger. And so this is potentially important, possibly to you now, because if you compress adipose tissue by like sitting on your butt for a while, the local stiffness that the adipocytes feel in this compressed tissue has risen by quite a lot. Several factors. And if you now do the same trick that I showed you before of taking cells out of the tissue and then culturing them on either soft or stiff polymers or hydrogels, if you take adipocytes and culture them on very soft materials, as soft as an unstressed piece of adipose tissue, they have a certain phenotype which, at least under the chemical conditions of a healthy, lean person, expresses very few inflammatory things and a lot of things that are good for adipocyte maintenance. But if you culture cells on materials that are as stiff as that of compressed fat, they very rapidly start to secrete inflammatory cytokines and become pro-proliferative. So compression, static and chronic compression of adipose tissue is well known to give rise to a lot of this stuff, a lot of inflammatory results, and if it happens because of the stiffening, it's potentially an explanation for why sedentary life and too much sitting is bad for you. So luckily, triggering this inflammatory response as a function of compressing a piece of tissue takes about 4 hours to happen. So if you sit down, you're probably better off getting up and having a cigarette or a candy bar than continuing to sit. So there is some evidence that this kind of local deformation has mechanical consequences.
Where does muscle sit on this?
So muscle would be much, much higher But if we compress, so I don't know about skeletal muscle. A heart muscle will more gently stiffen, and it has a much less dramatic response to compression.
See, I'm looking at this guy. You're trying to tell me it's unhealthy to sit on my butt, but what I'm wondering is, if I wear Spanx, does it make me seem like, my fat is muscular?
Oh, no, because the cell knows it's in the wrong environment. If there were muscle cells, they would be well maintained in a stiffer, more compressed environment. The readout of this mechanical thing, these mechanical signals, is really strongly cell type specific. So what's good or bad for one cell type is very different from another. Okay. Yeah.
The response to the compression is on a scale of hours?
Well, the inflammatory response, that's immediate. That's immediate. And I'm sorry, I forgot to show you this. As far as we can tell, it's a totally elastic, a recoverable response. So for example, if we take a piece of tissue and compress it and measure its shear modulus, and it's quite high, if we immediately decompress it, the shear modulus drops to where it was before we compressed it. So it is not because of a kind of a chronic remodeling of the tissue. It's whatever it is rapidly reversible. Okay. But we don't, yes? We glue it. We have to, these soft tissues we glue to metal plates with a biological glue, and so we can stretch it. Otherwise it will fail. Okay, so let me summarize this part. So I'm almost out of time, so I'm going to go to the next part pretty quickly. So I've shown you the wrong thing. Yeah. Biopolymer gels, as I showed you, stiffen as you increase the shear strain, but tissues don't. They soften. But what tissues do is that they stiffen with uniaxial compression. Not with stretch, but with compression. Okay, so now what do biopolymer networks do? Here's how we do the experiment. We put the thing between two plates, as I showed you before. But with biopolymer networks, as opposed to tissues, the mesh size is big enough so fluid could in principle leave or enter the sample as we compress or extend them. The tissues can't. They have these cell membranes everywhere. We never change the volume of the sample when we compress it. It just bulges out at the edge. But the polymer networks might. And so here's what happens. Here's just a boring control experiment, just to show that we're not introducing terrible artifacts from this very nonlinear way of doing the mechanical measurements. If we put into it a polyacrylamide gel, a linear elastic thing that's very well understood physically, we get exactly what we expect. The shear modulus in red symbols is completely insensitive to the axial strain, either in stretch or compression. So these linear elastic things they shouldn't care whether you stretch them or compress them. They don't. Shear modulus is independent of that. And if we look at the axial stress, how much resistance there is to compressing or stretching it, you get this beautiful straight line. The slope of the straight line should be the Young's modulus E. The Young's modulus E is about 3 times the shear modulus, exactly what we expect if the Poisson's ratio is about 1/2. So this is a pretty well-controlled, boring hydrogel. But these nonlinear hydrogels are really different. I'm going to skip over the dynamics for a second and just show you the result. If you do the same thing with a biopolymer network, you get this peculiar result. If we first look at the shear modulus has, again, a very asymmetric response to axial compression or extension, but it's exactly opposite what tissues do. If you take something like a fibrin network or a collagen network, or by extension maybe a cytoskeletal network, if you compress it and measure its shear modulus, it gets smaller and smaller. Not a lot, but like a factor of 2. If you stretch it, the shear modulus goes way up by like a factor of 10. And again, this is totally reversible. If we change the axial strain and remeasure the shear modulus, this will immediately drop down, this will come back up.
So just to make sure I understand, shear modulus is doing this?
Yes.
But then you're like stretching it this way and doing this?
Yes, yeah.
Compressing it this way and doing this?
Exactly, yeah. So we're statically keeping it stretched or compressed and then doing this. to try to get a handle of what happens in a real tissue, because most tissues are either stretched a little bit or compressed a little bit. And the cells within them are living in that static, compressed, or stretched environment. So the change in the local shear modulus we see by stretching and compression is not mimicked by these polymer networks. In fact, they do exactly the opposite. And if you look at now the axial stress, it has two modes. It has a very soft mode in compression. It has a much stiffer mode in extension. So networks like this are easy to compress, difficult to stretch. This is work of Van Oosten. It's again not specific to any chemical system. If we do it with a completely different protein, but something that has the same generic physical features, we get the same result. Collagen networks are very soft to compression, very stiff to extension, and the shear modulus goes all over the place depending on whether it gets uniaxial stress or compression. So this is interesting, and I don't have time to go over the theory, but just in case some of you are interested, Fred McIntosh's group has been computing these things from simulations of networks where we can put in the physical features of polymers like fibrin and collagen, and they've now done simulations in 2D and 3D of networks like this, and they get exactly the same generic features. That networks that have this feature, that they're made out of open meshes of polymers whose persistence length is on the order of the cross-link distance, so relatively stiff. They are soft to compression, presumably because filaments can buckle. They are very stiff to extension for a variety of reasons, which I'm not sure that I understand fully.
If you compress a rope, you have a rope, a string, you can push the ends together,
Yeah.
but you can't pull them apart.
Yeah. There's something very similar to that. But if you made a network out of, and maybe it has something to do with kind of a 3D version of that. What's a little more hard to understand is why the shear modulus cares so much about what happens to the axial tension or compression of these objects. Okay, so let me skip this and just get to the part that I think is more interesting. So for these semi-flexible polymer networks, the generic kind of feel to them is that they have a lot of poroelastic relaxation, which I glossed over, but that has to happen before we do these static elastic measurements. But they seem to have two different Young's moduli. They have a Young's modulus for compression, which is small, and a Young's modulus in extension, which is large. And that if you look at the shear deformation, it softens in compression and stiffens in extension, sort of in parallel, not exactly in parallel, but at least related to what the Young's modulus, or what the extensional modulus does. And so the question is, why are these networks so different from tissues? Why can't we explain the tissue response with these network phenomena? And there are two possibilities. So here's how different they are. If we look at, again, just how the shear modulus scales with compression, polyacrylamide doesn't scale at all, it's just constant. These open meshworks decrease. Tissues all get stiffer and stiffer the more we compress them. Okay, so there are two obvious reasons why that might be done, might be the case. We've looked at it by looking at blood clots. So blood clots are kind of a poor man's, simplified model of tissues. They're open polymer networks, like an extracellular matrix, populated with cells. But we can do a lot to change the relative contribution of cell to matrix in a blood clot. So blood clots have these two interesting features. About 40% of the volume is taken up by red cells, volume-conserving particles that are sitting in this polymer network. But they also have these platelets, which put internal stress on the network. And both of these happen in tissues. And so we've asked, could the difference between tissues and polymer networks, is it related to the internal stress that cells put on the network, or is it related to the fact that there are so many particles embedded in the network? And so blood clots allow us to do this. So Here's the blood clot again. It looks very much like a naked fibrin network. Here's the polymer by itself, which is easy to study. It's the same polymer network, except turquoise now. And in a real blood clot, it's full of these contractile and non-contractile cells. What's nice about blood clots is that, well, what's different from a blood clot from a tissue is that the volume fraction of the cells is only like 40 or 50%. In a tissue, they're pretty much close packed. And the tissue is more like a close packed bunch of spheres through which the polymer network penetrates. A blood clot is like a polymer network which is decorated with some particles. But we can convert the blood clot to something more tissue-like by allowing the platelets to continue to do their job and just squeeze all the extra fluid out until the network collapses around the red cells, and now we have completely packed red cells under some tension. And also we can put poisons to prevent the platelets from doing their thing, and you can sort of see here that once the blood clot has retracted, the red cells are packed together, and it's sort of more like they're kind of decorated with polymers rather than being inclusions inside a big polymer network. Okay, and here's what happens rheologically. If we take this complicated blood clot and simply poison the internal pre-stress that the platelets put on, the blood clot pretty much just looks like a naked fibrin network. So there's almost no signature of the fact that most of the volume is occupied by cells. The whole rheology, it's again softens in compression, stiffens and stretch, just like a naked fibrin network does. The presence of these particles does almost nothing to it. So it's not the simple presence of particles that really changes the material properties. What begins to change it a little bit is the pre-stress. If the cells, the platelets are now not poisoned, but they're allowed to put contraction onto the networks, then some things begin to change. Again, here's the fibrin network, these two different Young's moduli, this continuously changing shear modulus. Now we get rid of this two different Young's moduli. Once we pull all the slack out of the actin filament, or out of the fibrin filaments, we begin to lose this kind of soften compression, stiff and extension. Now it's a pretty linear, elastic thing. But the shear modulus is still softening in compression, stiffening in extension. So it's not simply pre-stress in the network that causes a polymer network to look like a tissue, nor is it simply the red cells. However, if we now let all the free volume get squeezed out of this blood clot until the red cells touch each other, it's still exactly the same material. We've just increased the volume fraction of particles inside the polymer network. That's what the retracted clot is. Now the thing stiffens in compression rather than softens. So all it takes is either internal pre-stress, or if we could do it magically, fill a polymer network with close packed particles, something we technically have not succeeded in doing yet, we may be able to get these kinds of compression stiffening materials. So this is at least a signature that simply looking at tissues from this kind of polymer network point of view has really big limitations. We have to pay much more attention to the stuff inside. So let me just conclude here just to remind you, the networks of cross-linked stiff polymers have this really complicated relation between the stiffness in the axial direction and the shear stiffness. Somehow those two things are coupled to each other in a very strong way, which is barely beginning to be understood. So what happens to the networks is that they soften in compression, stiffen in extension. Soft tissues do exactly the opposite. They stiffen in compression, they soften in extension. And that this conversion from this kind of model to this kind of model, at least in the blood clot sort of analogy, happens as soon as the particles inside begin to touch each other. Okay, let me stop there, and thanks for your attention.
So I have the first question. Oh, you go.
Is there a cell addition here in that? Because I would think
Yeah.
it does compare to like
Yeah. So in a tissue, it probably would. In the blood clot, it doesn't, because the red cells have no connection to each other. The platelets do, but they're kind of negligible. But the cell-cell adhesion must play some role, but it can't be dominant because fat, for example, there is no cell-cell adhesion in fat. Every fat cell is surrounded by a little thin cage of extracellular matrix. The cell does adhere to the extracellular matrix, yes. And, oh, sorry, let me take that back. We have some evidence that cell-cell adhesion, and certainly that cell matrix adhesion, is very critical to this stiffening of tissues, because it's possible in the liver, for example, to perfuse the liver with what turns out to be a snake venom that destabilizes the cell matrix attachments. And when you do that to the liver, you do not change the uncompressed shear modulus, but you very strongly eliminate compression stiffening. So as soon as you detach the cell from the cage around it, the physics changes, the mechanical response changes quite a bit. Yeah.
If I put a stem cell on a 10 kilopascal substrate, does it become muscle?
Not by itself. What the stem cell will do will become, if you don't give it the right chemicals, it will become as stiff as, it'll become 10 kilopascal. It will still be stem-like, but once it gets to that state, you know, Dennis Disher and other people have shown that cell, depending on what kind of stem cell it is, but that stem cell will now become acutely responsive to chemical cues to become a muscle. And it will be very unresponsive to chemical cues to become fat. But if you put the same stem cell on something that's very soft, the stem cell will be soft, and now it won't listen to signals to become a muscle. It will prefer to become a fat cell. But it needs both. It needs the mechanical thing, maybe to prime it, and then it becomes more or less susceptible to the chemical signals.
This is maybe in the same direction as the last question, but so you showed that the way the cell acts really depends on the stiffness of the substrate. Why is that? Why is it just rather?
Yeah, so that's not super well understood. In fact, it's not hardly understood at all. There are a couple of hints. One is that there is quite a bit of evidence that because the cell is always putting tension on whatever it sticks to, the act of sticking apparently often engages actomyosin contractility. And depending on how much resistance it feels, that can either unfold signaling proteins inside or open calcium channels and start to drive chemical signaling. And one of those chemical, if you keep it cultured for a long time on stiff or soft substrates, that can lead to transcription regulators turning on a bunch of transcriptional programs. So that part is really, really very little understood or studied yet. But I think the idea is that as soon as it sits on the surface, it starts pulling on it. That's almost an automatic response. And the mechanical consequence of that, of applying attention to that adhesive thing, can lead to different effects depending on whether the inside of the cell is stiffer than the outside of the cell.
Is it like saying it's too stiff here and then I'm going to try to change the environment in some way? Is that like ultimately what's going on?
I don't, well, unclear. And it's also not, it's also, I should say, it's also not clear that cells always become bigger or somehow happier on stiff materials. There are lots of cells that grow better on soft materials than stiff materials. More of them probably look more proliferative. And it's also, so how that mechanical signal from the substrate stiffness couples to biochemical signal is sort of way in its infancy. But presumably it's working. There's no reason it couldn't work just like chemical signals. You have a transmembrane protein that does something in response to a force. And once it responds, it can send the same kind of activate tyrosine kinases or whatever it does. That could be totally the same.
Is it possible, or has anyone done the measurement, if the amount of cell to matrix or cell-to-cell adhesion depends on the compression?
Yeah, oh, good question. I don't know that people have looked at that much. It's been looked at, I think, a little bit in like embryogenesis. I mean, people have certainly taken these like spheroids of cells that are collections of tens or hundreds of cells and put uniaxial compression on them and watch them move past each other. And so a lot of that has to do with how many of the adhesive proteins that the cells express. But the fact that you sort of change the cell shape by compressing it, we haven't looked. That's actually a good question. I think it's not, unless somebody knows. I don't know the answer to that. I don't think it's been really looked at.
Okay,
Yes.
I'll ask a question about how does this system compare with just a packing of, say, soft spheres? Because if you compress those, you're basically storing the
Yeah.
energy in the deformations.
Yep.
And so, of course, the bulk modulus will increase, and as you
Yeah.
shear it, the shear modulus
Yeah.
increases too. So this is sort of known as the
Yeah.
anomalous viscoelastic response of jammed matter,
Yeah.
because the bulk and the shear modulus grow, but the bulk modulus
Yes.
grows. It appears to be very stiff,
Yeah.
very abruptly when the system reaches jam.
Yeah.
So there seems to be a discontinuity in the response, and I just
Yeah.
wondered, is that, but at least qualitatively the response would be similar.
Yeah.
It's that as you compress, the shear
Yeah.
modulus also goes up.
Yeah, I think that's a really likely explanation. There's something there, but it is not. What's kind of interesting is it's never abrupt here, and it's always smooth and continuous. So what we're working on and have not done is just a kind of a graded response. In blood clots, or if we put in swellable particles or something, we don't have a good measure yet of what the shear modulus is as a function of packing density, for example. We don't have that number. What we know is that if we take these things and compress them more and more, we never see soft, 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 and then suddenly something takes off. And so it could be.
You don't see a jamming transition in.
We don't see, and so I wonder if whether some of that is hidden by the fact that it's a soft particle system that's embedded already in an elastic network. So the elastic network is always there, and so we don't. It's also for the same reason when we stretch it, and we would go way below the jamming transition, we still have a good shear modulus. So that's coming from somewhere, and presumably that may be blunted out by the fact that we have an elastic network within which these particles are embedded.
Yeah. Any more questions? If not, we can continue chatting upstairs on the 6th floor. There's 9 machines as usual.